Hi guys, welcome to my YouTube channel. Today's video is going to be on animal and plant cells. We're going to go into a little bit more detail about the different organelles that are found in both the plant and the animal cell. These are concepts that you'll be expected to know, and we won't be going through these exact same slides in class, so make sure that you are copying down notes. And just remember, since this is a video, you can definitely pause because I will be going a little bit, I'll be going at a faster rate when I'm speaking. So just pause, jot down the notes, and then press play to continue the video. So enjoy, and I'll see you in class tomorrow. So there is some terminology that you do need to know, and you can get easily confused by some of the terms because they seem very similar to each other. So ECF is the extracellular fluid, and that is the watery environment that's outside the cell. So all of the fluid that's outside of the cell, that's considered ECF. Cytosol, on the other hand, is the aqueous solution that's inside of the cell where all of the organelles are suspended in. So where all the organelles are, that aqueous solution that's in the cell, that is called the cytosol. Whereas the cytoplasm is just a general name that's given to both the cytosol and the cell organelles that are outside of the nucleus. Your cell membrane, if you recall, your cell membrane is what controls what goes in and out of your cell. And it's special because it's made up of a phospholipid bilayer. A phospholipid is a fat molecule that's in a cell membrane, and that specific membrane is made up of a bilayer, so it's a double layer of fat molecules. So if you're looking at the two pictures that are below, the first image is of a cell, and there you see like all those little circles. All those yellow circles are all these different phospholipids, and if you look at the picture that's in the blue, what you'll notice is that every single phospholipid has a hydrophilic head and a hydrophobic tail, and they're kind of layered together in a double layer to create that cell membrane, and that is what controls what can go in and out of the cell. So just a question for you that I will ask tomorrow in class. Why do you think that the cell membrane has a phospholipid bilayer? As in, why is it not just one layer of phospholipids? Right? So if you're looking at the image below, that there is of a cell, and what you'll notice is that the cell membrane is made up of that phospholipid bilayer, and each phospholipid consists of a hydrophilic head and a hydrophobic tail. So think about what the answer could be, and we will discuss that further in class tomorrow. In terms of this comic, hopefully you are able to understand why this is moderately humorous. It's called Membrane Pranks, and there you've got the phospholipid, and he's saying, hey guys, look, I've got a water gun. How is that funny? I'll ask you again tomorrow. So moving on to the nucleus, if you remember, the nucleus is the control center of the cell, so it determines all of the cell's activities. And like the cell membrane, the nucleus also has a double membrane, so it has a bilayer of phospholipids. This here is a micrograph, so a photograph taken using a scanning electron microscope. And what you can see there is a nuclear envelope. Your nuclear envelope is what surrounds the nucleus, and that is filled with nucleoplasm, that which contains RNA and DNA. So if you're looking at the picture, there you see in purple, the light purple would be the nuclear envelope, and that's surrounding the nucleus, and all of the fluid that's within that nucleus is that nucleoplasm, and within your nucleoplasm you've got your RNA, your DNA, and your nucleolus, which is that darkest part in the center of your nucleus. So the nucleolus is the dark structure that's inside the nucleus. It's where all of the ribosomes are formed. Your ribosomes, if you're wondering what they are, right, you should hopefully have a good understanding of what ribosomes are from the basic information. But ribosomes, they're located everywhere in the cell, and they're attached to the RER, which is the rough endoplasmic reticulum in the cytosol. And a good question to ask would be, oh, why is it that ribosomes are located everywhere in the cell? So not only are they attached to the RER, but they're also found scattered within the cytosol. Ribosomes are used by the cell to make proteins, and so all the free-floating ones within the cell make protein for the cell, and then the ones that are on the RER make protein for use outside of the cell. So there is rough and smooth endoplasmic reticulum. So if you're wondering, like, what's the difference between the two, RER would be rough endoplasmic reticulum, and that has attached ribosomes that make protein, whereas your SER stands for smooth endoplasmic reticulum, and that has no ribosomes, so it's not studded with all the ribosomes, it's just smooth, and it makes molecules of fat. Your Golgi apparatus, some say it looks like a flattened set of balloons. Your Golgi apparatus, as you know, it packages and ships, right? So what it does is it changes the fat and the protein that's made by the endoplasmic reticulum, packages them into little vesicles, and then ships them out to the ECF. So it ships out all of these little vesicles out into the extracellular fluid. So it ships it out of the cell, like through the cell membrane into all of the extracellular fluid, so fluid outside of the cell. And that's why they say your Golgi apparatus is kind of like shipping, right? Because it packages and it ships, so it's like UPS. Your lysosomes, they are found only in animal cells. They contain protein and can be used to digest food particles brought into the cell from the extracellular fluid. So your lysosomes, they contain protein and they can digest food particles brought from outside of the cell. And what's really good about them is that they can destroy dangerous bacteria and viruses. And one really interesting thing is that about lysosomes is that cells are actually suicidal. So when an animal cell gets old, those lysosomes, they break open and they decompose the entire cell, and that's called apoptosis, which is cell death. And so cell death is just a routine, healthy, and essential part of life. It's how babies develop fingers and toes and how tadpoles lose their tails. Your mitochondria, if you recall, is what supplies a lot of the energy, right? It's kind of like the powerhouse of the cell. It creates energy for the cell, and it's mainly responsible for cellular respiration. So cellular respiration is just a reaction that produces compounds that cells use as energy. So it's the basic equation is sugar and oxygen will then produce carbon dioxide, water, and energy. Your plastids. Plastids are just organelles that are found in the cytosol of plant cells. So plastids are unique to just plant cells. You won't find plastids in animal cells. Your chloroplast is a special type of plastid, and that is found only in plants, and it's responsible for photosynthesis. And the key thing to make note of in the picture below is of a chloroplast, and you'll see that your chloroplast is made up of thylakoids and granum. And then the fluid within that chloroplast is called the stroma. Right? So your thylakoid are like each of the little coins, and then the granum would be a bunch of the coins stacked together. And then the stroma is the fluid that's found within your chloroplast. Chromoplast is another type of plastid. It is another cell organelle that's found only in plants, and it's responsible for all the orange, yellow, and red pigments found in things like carrots, peaches, and corn. And last but not least, we've got the amyloplasts. Amyloplasts are cell organelles found only in plants, another type of plastid. This time it's non-pigmented, so chloroplasts are pigmented green, chromoplasts are either yellow, red, or orange, and amyloplasts have no pigment at all. And what they do is they synthesize and they store starch. So one really interesting thing is that they can actually turn into chloroplasts. So for example, when you've got your potatoes, right, you've got your potato tubers, when they're exposed to light, they turn green. And similarly, chloroplasts can turn back into amyloplasts. So if a chloroplast accumulates too much starch, then it will become an amyloplast. So they kind of switch back and forth from each other, amyloplast to chloroplast, depending on how much light and how much starch it's made up of. And the last question that I have for you would be about vacuoles. So as you know, vacuoles are found in both plants and animal cells. But the question I pose is that vacuoles in plant cells are bigger than in animal cells. And why is that? So why is it that vacuoles in plant cells are bigger than those found in an animal cell? And maybe this image here could be a little bit of a hint, and we will go through it together in class tomorrow.